Okay, I think we should make a start. It's just gone after 10 o'clock. Good morning, good afternoon, and good evening, wherever you happen to be around the globe. It's my pleasure to welcome you to the third webinar of the winners of the Sheikh Hamdan International Awards. My name is Gulfaraz Khan. I'm Professor of Viral Pathology at the College of Medicine and Health Sciences, UAE University within the United Arab Emirates, and member of the Sheikh Hamdan Awards Committee. Before I introduce our speaker, Professor Ian Fraser, the winner of the Grand Hamdan International Award, I would like to say a few words regarding the awards itself. The award was established in 1999 by His Highness the late Sheikh Hamdan bin Rashid Al Maktoum, with an objective of honoring scientists anywhere in the world whose research has significantly contributed to the larger interests of humanity. The award has several categories, the most prestigious of which is the Grand Hamdan International Award. Some previous recipients of this award have gone on to win the Nobel Prize. The latest example of this is Dr. Harvey Alter, pictured here. He was awarded the Sheikh Hamdan Award in 2016 for his contributions to the discovery of hepatitis C virus. And he later went on to win the Nobel Prize in 2020. Now, in the latest cycle of the Grand Hamdan Awards ceremony took place on the 23rd of November 2022. And the winner of the Grand International Award was Professor Ian Fraser. Ian Fraser is Emeritus Professor of Medicine at the University of Queensland and the Chair of Translational Research Institute. He graduated in medicine from the University of Edinburgh and trained as a nephrologist and a clinical immunologist before moving to Australia. Ian's research has basically focused on human papillomavirus and vaccine development. He has published hundreds of papers in high impact journals, and he has dozens of patents to his credit. He heads or chairs numerous organizations and biotech companies. Just to give you a couple of examples, he was the inaugural president of the Australian Academy of Health and Medical Sciences. He chaired the Australian Medical Research Advisory Board. He was advisor to the WHO and the Bill Gates Foundation on HPV vaccines. Ian has received numerous awards, both at national and at international level. He was given the Australian of the Year Award. He was awarded the William Colley Award together with Harold Zuhausen, who also won the Nobel Prize in 2016 or 2018, I believe it was. He also was made Companion of the Order of Australia by the late Queen Elizabeth II of UK. Ian has also been elected to the Fellow of the Royal Society, both of London and Edinburgh. And the latest recognition is he was awarded the Grand Hamdan International Award in end of 2022. Now, before I pass the floor on to Ian, a couple of housekeeping points to bear in mind for the audience. There will be a questions and answers session at the end of Dr. Fraser's seminar. Please post your questions in the chat box, and you can do that as you go along. Please make your questions direct and brief. The objective is that we want to try to get through as many questions as possible. Certificates will be available, so all the attendees who registered, there will be certificates available, and you can download these by end of tomorrow. So the certificate, all of you who have registered will be able to download these. That's all from my side. Dr. Ian, the floor is yours. I will just stop sharing my slides and okay.
Okay, I hope you can now see my slides, and I'd like to thank Professor Khan for the kind introduction. He makes more of me than I actually am, I think. What I'm going to do today is to talk about cervical cancer, first of all, from the point of view of prevention through using the immune system, and then secondly, as a model for immunotherapy for cancer. Cervical cancer is a very significant problem globally. Approximately 500,000 women will acquire cervical cancer every year, and there are approximately 300,000 deaths from this disease. So what I'm going to do is to talk a little bit about how we should be preventing this rather unfortunate statistic. The story of prevention of cervical cancer started quite a long time ago, nearly 100 years ago, when George Papanikolaou developed a treatment, a screening test for cervical cancer by taking samples of cells from the neck of the womb of women. And this was a very major step forward. He first described this in 1928, and as you can see in the slide here, you can imagine that these are normal epithelial cells from the cervix. These are the precancer ones, which obviously we're trying to identify to enable treatment. And he published a book about this in 1943, but I have to say that it was quite a long time after that before this test had a global impact, because until it became routine for all women to get screened for cervical cancer, the incidence of cervical cancer globally did not fall. And it was only in the 1980s, for example, in the United Kingdom, with better coverage of screening, that we could actually see a reduction in cervical cancer. Of course, this gentleman, Harold Sirhausen, was a very important contributor to the knowledge base on cervical cancer, because he was the one who first postulated that perhaps a virus might be responsible for cervical cancer. He was interested in the genital warts and knew that there were papillomaviruses that were responsible for these, but he hypothesized that it might be possible that also cervical cancer would be caused by viruses of the papillomavirus family. And his colleagues, and particularly Lutz Gisman and Andreas Kleinherz, went on using the then absolutely novel technique of southern blot hybridization to show that there were papillomavirus DNA in the not only in the cervix of women with cervical cancer, but also in the cervical cancers. And this was a very major step forward in understanding why this disease came about. We, and when I say we, are my colleague, Dr. Jan Zur, and I, thought that this would give us an opportunity for prevention of cervical cancer by developing a vaccine, and I'll talk a little bit more about that in a moment. It also gives a possibility of specific immunotherapy for cervical cancer by attacking cells that express human papillomavirus, and I'll talk about that a bit further too. We started off by expressing the papillomavirus capsid proteins and using vaccinia virus, and we developed these virus-like particles which look like the virus down an electron microscope but are not actually infectious. And these then became the basis of a potential vaccine for cervical cancer. which was developed in the 1980s with my colleague, Dr. Jan Zhu, shown here, and the virus-like particles that he and I developed, which we went on to develop using insect cells and baculovirus. And he was very much helped by his wife, Xiaoyi Sun. Jan passed away about 20 years ago, but his wife is still around and still very much an advocate for his research work. That discovery of the virus-like particles took about 15 years to turn into a product, Gardasil and Cervarix, which basically were shown to be effective in clinical trials over a longish period of time. And these are the basis of the vaccines that are now currently available globally to prevent cervical cancer. The trials that showed that cervical cancer vaccines were effective were very large trials, run globally. The reason for that is, of course, that for all the people that get HPV infection, only 1 in 100 will go on to develop a cervical precancer. And the studies were required to show that cervical precancer could be prevented by the vaccine. It wasn't sufficient simply to show that papillomaviruses could be prevented from infecting the cervix. It was necessary to show that there was actually prevention of cancer as well. And so these 5 studies are typical of the studies that were done, and they all showed nearly 100% effectiveness against papillomavirus, but with 2 preconditions. One was that they were not effective if somebody had already had the infection. The vaccines only work for people who have not yet seen the virus. And secondly, that the vaccines were only effective against the HPV types that were included in them. The initial vaccines included type 16 and 18, which together were responsible for about 70% of cervical cancer. The vaccines that are now available also include 31, 33, and 35 types. And these together collectively are about responsible for 95% of cervical cancer. So the vaccine programs run globally now should be effective at controlling cervical cancer. How then are we doing with control of cervical cancer globally? Well, we've delivered 400 million doses of vaccine globally to date, to the best of our knowledge. And we know that there are 25 million women out there today who will die of an HPV-induced cancer if they have not been vaccinated. In principle, the vaccines that have been delivered so far should save approximately 4 million lives. 1 in 100 people who get the virus will go on to get cancer. But unfortunately, in practice, it's a much smaller number because most of the people who have received the vaccine are also taking part in screening programs, which are widely used in the developed world to help prevent cervical cancer through the Pap smear test that was invented by George Papanikolaou. And so the major problem now is to get these vaccines where they're actually needed in the developing world. This map basically shows the extent of the problem. The countries that are coloured in green are the countries which have got extensive vaccine programs to help prevent cervical cancer, and the countries that are coloured in purple are where most of the cervical cancer is actually seen. And so we have this challenge of getting the vaccine in the countries that really need it. So we're doing some studies to demonstrate that there is no major block to vaccine delivery so long as vaccine is made available. And we chose Vanuatu as the first country. It's an island near to Australia where I live, and it has a very minimal health system. It's got 250,000 people, but only 5 doctors and only 1 vaccine fridge for the whole islands. So that we wanted to demonstrate that we could effectively deliver vaccine there. We went out and screened 500 women over the age of 30 in Port Vila, the capital city there, and found that 500 of these 500 supposedly well women already had cervical cancer, and another 40 of them were in the process of developing it. So this gives you some idea of the scope of the problem there. Fortunately, we were able to set in place education programs in Vanuatu. Here in the picture, you can see the district nurse informing the children at school and their parents and also other district nurses about the vaccine program. And we ran an education program for a couple of years before we started actually vaccinating there. When the vaccine program was rolled out, We chose one of the islands in Vanuatu, the main island, which is called Ifati, where Port Vila, the capital city, is. And we managed to get 80% of the 10 to 12-year-old girls that were eligible for vaccine to take part in the vaccine program. When I say we, it was really the district nurses there that did the program. We only came along to give them some advice. It was very important that they were the ones responsible for delivering the program. And of the ones who got the first dose of vaccine, we managed to get virtually all of them to get the second and third dose. We don't recommend a third dose now, but the second and third dose was recommended at that time, which was very similar to the figures that we got in Australia, where there was plenty of education about the vaccine and an enthusiasm for preventing cervical cancer was found across the whole profession, medical profession there. So this was a very encouraging result. That it said that very clearly that if we could give the country a free vaccine, then they would deliver it effectively. We went on to convince ourselves that was true by choosing another country where, again, there's limited medical services, and this was Bhutan. Bhutan is a very poor country, but they have a very willing royal grandmother who was keen to see her country protected against cervical cancer. So we went to visit them and taught them about the vaccine. And the royal grandmother, she told the two gentlemen with the saffron robes, the Minister for Health and the Minister for Education, that she wanted a vaccine program for her country. This was received the blessing of the church there. This is them actually blessing the vaccine in the box here. And what we found was that if we delivered the vaccine through schools, we could get nearly 100% coverage of eligible girls. Schools programs were difficult to deliver because the schools are often a long distance from the nearest road, so that they tried delivering the vaccine the other way around, asking the school children to walk to the health centres, which generally were on the roads, and there was a rapid drop in vaccine uptake. So now the vaccine program there is universally delivered through schools. So in principle, we should be able to eradicate cervical cancer, and Dr. Tedros, who is the WHO director, has made this his challenge during his time in office, is to develop vaccine programs to eradicate cervical cancer. However, it's not that easy, because these are data from Australia where we've achieved a very high rate of vaccination. What this really shows is that even although we have vaccinated all girls aged 12 to 14 as they come through schools, and we have managed to do that now for over 10 years, the reality is it will take nearly another 40 to 50 years before we can reduce the incidence of cervical cancer down to an acceptable minimum level. And that only works if we carry on the screening program as well. If we don't carry on the screening program, there will be a steady rate of cervical cancer right up to the end of this decade and century. So this is the big challenge, and therefore we have to think also about whether we can use our knowledge about papillomavirus and cervical cancer to actually effect a means of treating cervical cancer through vaccination. So that's what I'm going to talk about for the rest of the talk. And hopefully this will be of interest to those of you who are interested in immunotherapy more general. So treating HPV infection with virus-specific immunotherapy, as I've said, is needed even if a prophylactic vaccine works. The challenge with that, of course, is that we haven't yet managed to develop many vaccines which are effective for treating existing infections. So what do we know about immunotherapy? Do we really know that we can actually achieve things by using the immune system for cancer? The answer, surprisingly, is yes, from some experiments that were done a very long time ago. William Coley was a surgeon in New York in the 1890s, and he wanted to try something different for treating cancers. And what he did was he used bacterial products to stimulate the immune system of the patients that he was treating with cancer. These bacterial products were what we would now call cytokine inducers. And with the cytokine inducers that he gave to the patients, he found that he could get a 40% cure rate of the patients he selected for treatment. He selected those patients that had sarcomas, and they were perhaps more susceptible to this sort of treatment. I don't think you get a 40% cure rate for the bacterial products that he was promoting at that time for all cancers. so that it's at least an example that in fact some sort of progress can be made against cervical cancer by using immunotherapy. So immunotherapy was actually not the newest form of cancer treatment. It was actually the second newest after surgery, but it came before both radiotherapy, which Madame Curie promoted in the 1920s, and chemotherapy, which only was first used in the 1950s. So the immunotherapy for cancer has actually been around for a long time and has a good track record. So what we need to talk a little bit about today is how should immunotherapy work and why it doesn't always work, and what we can do about that, particularly in relation to cervical cancer. So Sir Macfarlane Burnet, who was an Australian scientist who was particularly interested in flu vaccine, but also very much interested in the immune system, he proposed the concept that it should be possible to treat cancer with immunotherapy. He observed that immune surveillance prevents some tumors in animal models, and most importantly, also went on to show that tumors have specific antigens that are seen as not self by the immune system. These can, of course, be viral gene products like papillomavirus gene products, but there are also regulatory genes in cancer cells which are mutated and therefore can be seen by the immune system. And antibodies to tumor-specific antigens are common in cancer patients. That, of course, doesn't mean that the cancer patients are making an effective immune response. What's actually happening is that the wrong sort of immune response is being generated. Antibodies are not thought to be likely curative for cancer. Rather, we need killer T cells. And killer T cells require the induction of an appropriate immune response, which basically it requires a certain sort of inflammation to produce the right sort of killer cells. And what I'm going to do for the rest of the talk is talk a little bit about what we've learned about that. But what we do know is that we can use the newer technologies that we now have available to map out why there are problems with cancer immunotherapy. We also have to recognize that the tumors can fight back against the immune system. Tumors mutate to produce more effective clones that will grow in the host, even despite the treatments that we use. And of course, that includes immune treatments, so that there are also a requirement for controlling the problems of immune inhibition in a patient that has cancer. The biggest proof of all that we actually naturally make an immune response to cancer is to look at what happens to the incidence of cancer when patients are immunosuppressed. And the group of patients that we can use for assessing this issue are patients that have received a renal transplant, because they are all given drugs to suppress their immune system while they've got a kidney transplant on board. And what this slide shows you is a whole list of different sorts of cancers. And if the dots for that cancer lie to the right of the red line here, that means that these cancers become more common with immunosuppression. In other words, under normal circumstances, the immune system helps to control them. And that you can see that a large number of these cancers are cancers which are associated with papillomavirus infection. So that, for example, obviously cervix cancer is on the list, but also vulval and vagina cancer and esophageal cancer, which are all common cancers, which we now recognize are associated with papillomavirus infection. Interestingly, there are a whole range of cancers which are not thought to be associated with viral infection, where again, immunosuppression increases the risk of developing the cancer, particularly kidney and thyroid cancer. But many common cancers are slightly more common if your immune system isn't working, suggesting that the immune system can do something to help prevent these cancers as well. So we're focusing particularly on the papillomavirus associated cancers, because that's where I'm particularly interested for my research. So we know that the papillomaviruses that are responsible for cervical cancer do so by producing two specific proteins called E6 and E7. These are not viral structural proteins. These are proteins which impact on the way that cells that are infected with the virus replicate. And if you like, these two proteins together drive continued replication of the epithelial cells that papillomavirus infects. So that's been known for quite a while, and it's the basis of much of the work that's been done about vaccines to help treat patients that have got cervical or other papillomavirus-associated cancers. So back in the 1990s, we thought that it would be a good idea to try to develop a vaccine that was focused on producing an immune response against the E6 and E7 proteins of papillomavirus, and in specific induce cytotoxic T cells that could therefore potentially kill cancers that were expressing these proteins. We used an adjuvant which at that time was well shown to be both safe in humans and also a very strong stimulant of T cell production. It's a saponin from the Quilea plant, and it's called Discomatrix. And we started a trial using these proteins. Before we went ahead to do the trial in humans, we demonstrated in an animal model that this is a very effective means of producing an immune response against papillomavirus. We used a transplantable tumor called TC1, which expresses the E7 protein of HPV16 obligatorily. And indeed, that protein drives these tumors to grow in mice. And what we did was we immunized the mice when there was a well-established tumor in them with the E7 protein of HPV16 and our vaccine Discomatrix. And what we observed was that with this combination of Quilea saponins and E7 protein, we could cure 20 out of 20 mice, whereas if we just gave them a saline buffer, all the mice succumbed to the cancer. So we thought this was a fair model for working out if cervical cancer could be controlled by papillomavirus. We went on, therefore, to do a trial in humans, women with cervical precancer that were going to be treated surgically anyway. And what we did was we immunized them with the papillomavirus E7 protein using Quilea saponin as the adjuvant to see if we could eradicate their cervical precancer. And we would find that out, of course, by doing surgery on the cervix to check and see if we had cleared it. What we found was that our vaccine in humans produced a very nice immune response. We could measure antibody, which we weren't really interested in, but we also got cytotoxic T cells. And this was enhanced particularly if we gave the largest dose of the vaccine in comparison with pre-immunization when there was no response to E7. There was a strong response to E7 with all of the assays that we did. Unfortunately, that was what happened. We got a good immune response, but we didn't get any effect on the cervical precancer. The CIN23 patients had CIN23 before they were immunized, and they still had it after they'd been immunized. So the vaccine produced a nice response in mice, but didn't work in humans. Not an uncommon problem for cancer immunotherapy. So we went back to our animal model and had another look at the data. And what we found was that we could show that while the vaccine was very effective in the mice, even the adjuvant on its own was quite effective at controlling the tumors. And so this was rather the equivalent of what Coley had found with his Coley's toxins, which were basically an adjuvant, that they were actually quite powerful in this animal model. But the model itself was not rigorous enough to allow us to develop effective vaccines for cervical cancer by using it as a test model. So we wanted a better model, and we ended up making a model in animals, which we think is a fairer model. So what we did was we took mice and we made them transgenic for the E7 protein of HPV16, driven off a keratin promoter so that the antigen was expressed only in the skin cells. Now, these mice, because they were expressing E7 from birth in their skin, were tolerant of E7. Proteins that are present in an animal at birth are tolerogenic. So what we did was we took a transplant of the skin from these E7 transgenic mice and put it onto a normal mouse. We kind of expected that that graft would be rejected by the mouse's immune system. We showed that the antigen was only expressed in the basal epithelial keratinocytes, and that we also showed that in this model we could find some antigens where the grafts were rejected. So if we made an over-transgenic animal expressing ovalbumin in the skin, then we could get the grafts to reject. But when we tried it with E7, the grafts were never rejected. But we could show that the E7 protein was expressed in the skin and that it induced epithelial proliferation, because that's what E7 protein does. It drives epithelial proliferation and eventually cancer development. And we also showed that the E7 transgenic skin had a heavy infiltrative immune cells. So this is a section through a normal mouse ear. And this is the epithelium here. It's quite narrow, and there's very little in the way of cellular infiltrate in the dermis there. This is a K14E7 mouse here, and you can see the epithelium is considerably thickened because the E7 protein drives epithelial proliferation. And in here, there's a strong inflammatory infiltrate, but obviously not the right sort to get rid of the skin graft. So we then decided that we needed to see if our model was a fair model of cervical cancer. So what we did was we looked at the gene signatures present in CIN3, so the cervical precancer in women, and compared it with the gene signature present in our K14E7 mice. And basically what you can see is that there's a very strong enrichment at this end of the scale. So what this is actually telling us is that the CIN3 in the cervix looks very like K14E7, E7 transgenic skin, and that we're basically induced a good model, at least in terms of the genes that are being expressed in the skin when compared with normal skin. So that it wasn't such a good model of cancer, but that didn't particularly surprise us because we didn't induce a cancer using our K14E7 mice, just the cervical precancer, the condition that drives to cancer eventually. So we thought about how we could try and understand why this grafts were not rejected. And we thought, well, maybe the problem is that there are regulatory T cells present inside the graft here that are stopping the immune system working. T effector cells, cytotoxic T cells, can be controlled by regulatory T cells, which are induced by the expression of the E7 in the skin. So we depleted regulatory T cells by two very standard methods. One was depleting out CD25 positive cells, and one was eliminating FOXP3 positive cells. And these are two mouse models that are available. And we looked to see if either of these models would allow us to get graft rejection. And the short answer was we did not get graft rejection with these. But interestingly enough, one more experiment that we set up was to set up a model where the graft had no immune system in it. So we put the graft expressing E7 onto a mouse with a normal immune system, but we took the graft from a mouse that had no T cell immune system at all. And then the grafts were rejected, as you can see here in the graph. So what that tells us is that the E7 protein is inducing locally in the skin graft an immune response which protects that graft against rejection. So we wanted to find out what mechanism was involved in achieving this. And so we did some experiments, and to cut a long story short, we looked at a lot of different sorts of immune response in the E7 transgenic skin. But the one that turned out to be important was a T cell called the NKT cell, because when we took a graft from an animal expressing E7, but with no NKT cells, and there are two different genetic models for producing no NKT cells in those mice, and put these grafts onto normal mice, the grafts were promptly rejected. So the NKT cells were inhibiting the rejection of the E7 expressing skin. And we went on to learn another very important factor from that was that if we put two grafts onto the mice, one of which had E7 in it and also no NKT cells, and the other was a graft which expressed E7, then this graft was rejected because it had no NKT cells, but this graft, which was competent for NKT cells, was not. So this was a locally inhibitory effect. And that meant that we had to do something locally to enhance the immune response to E7 if we wanted to get some effect against cervical cancer. And taking that one step further, we found that it wasn't just the NKT cells that were important, it was the interferon gamma being produced by the NKT cells that was important. Because if we did the same experiment, but now the only manipulation we did to the donor mouse, the mouse that was expressing E7 in its skin, was to knock out interferon gamma production, we also got graft rejection. So this says that this one molecule determines in the mouse model whether graft rejection occurs or doesn't. If there's interferon gamma produced in response to the proliferative epithelium, then you don't get rejection. If you stop the interferon gamma being produced, then you can get graft rejection. So this is beginning to tell us of what we need to be doing to make this work in humans. So that we wanted to see exactly what was going on in the K14E7 skin, and we used the technique of gene mapping to find out what was being expressed there. And what we found out was that there were these inhibitory molecules which regulate the immune response, PDL1, PDL2, and CTLA4. These are the molecules which now cancer immunotherapy specifically targets, so-called checkpoint molecules, which are controlled by checkpoint inhibitors. And we demonstrated that all of these were overexpressed in K14E7 skin in comparison with skin which expressed E7, but did not have the ability to produce interferon gamma. And then we also found that these were not expressed in normal mouse skin. So we've demonstrated basically that these are the molecules which are being induced by E7 expression and therefore potential targets for cervical precancer treatment. We went on to show more formally that there were a whole range of inhibitory molecules being produced. I won't go through the details of these in the interest of time. But perhaps more importantly, when we got the T cells out of the skin of humans that have got E7 in them and also the mice that have got E7, we could show that there was upregulation of PD1 on the T cells, that this is the inhibitory molecule that is induced by PDL1. And therefore, this was probably the mechanism that was stopping our vaccines from working. To take that one step further, we really wanted to see if we'd got the whole story. And when we had a colleague, Dr. Jenhausen, who came to work in my laboratory, and he basically took our grafting models as we did before and separated out the T cells that were induced in the E7 expressing skin. He wanted to see in what way they were regulated. And what he found out was that there was a very significant population of regulatory T cells and exhausted T cells induced in the skin of the K14E7 mice. And the effector T cells were present in normal numbers, but the helper T cells were of a regulatory type. And that they expressed phosphates-3 and interleukin-1 receptor 2. And this was perhaps a target now that we could start to think about using immunotherapy to eliminate. And when we went to do a little bit more exploration, we found no less than 6 different molecules which were basically there to inhibit the T cell function in the skin of these mice that express E7, and that we then went on to show that the similar proteins are expressed in the cervix of women that have CIN3. So we've gone round a complete circle. We've identified now what's inhibiting effective vaccines in patients with cervical precancer. We now need to find means of actually overcoming this problem. And basically, we can summarize the work by saying that proliferating epithelium is found in epithelial squamous cancer. It attracts regulatory T cells and alters the antigen-presenting cells, enhances checkpoint inhibitor expression, induces local immunomodulators. And therefore, we have several targets that we're going to have to overcome before we'll be able to get an effective immunotherapy for cervical precancer and cervical cancer in humans. So I'm just going to finish off with a very short story about something else, because this is I always like to put something new into any talk that I'm giving. And this is one of the problems that we have in Australia is another cancer, which very common is squamous skin cancer. This is just an example of squamous skin cancer. It's a cancer which is caused mostly by sunlight-induced damage to the genetic information in the skin. But what we found out was, somewhat to our surprise, that the bugs that grow on the skin of patients who have got skin cancer are different from the bugs that grow on normal healthy skin. And in particular, there's this huge abundance of staphylococci that grow there. Now, staphylococci are normally regarded pathogens, and they don't actually produce much in the way of local pathogenic effect in the skin directly. But what we found out was that they produce a very strong inflammatory immune response in the skin of patients with cervical precancer, and that this inflammatory response is present all the time when the staphylococci are growing on the skin. So that this is looking at the induction of these molecules by applying the staphylococci from skin onto normal skin, and we get this strong induction of interleukin-6, interleukin-8, and TNF-alpha. And more to the point, we could demonstrate that the staphylococci that were found on the precancer lesions, but not the staphylococci that were found in normal skins, produce a whole suite of toxic mitogens, in other words, proteins that make cells divide and also induce DNA damage. And this was found very consistently in the woman that we were screening for the squamous cell cancer of the skin. So that this gives us another set of targets that we can look for in the cervix, but also treat in patients. And we've gone on to do a preliminary study in patients with squamous skin cancer, where we treat potentially the staphylococcal infection with either Bactroban or muriposin, with some controls there to demonstrate whether we're getting a specific effect. I mentioned this because Jacoba Bromfield is my newest PhD student, and she's busily doing this work as we speak at the moment. So it only remains to point out that I've been describing work that's been a career of mine over 40 years now. And while I might be there in the background somewhere, all these young people here are the ones that are doing all the hard work to take this forward towards developing effective immunotherapy for cervical cancer. And I thank you very much for your attention. I'm happy to answer any questions that you might have.
Okay, Prof. Ian, thank you very much. Excellent presentation, and I'm sure there's lots of questions on some of the topics and areas you touched on. If I may take the opportunity to ask just a couple of, just to get the ball rolling. I know there's a lot of questions there. Can I just ask you one question with regards to the vaccine? You already in your presentation indicated that some of the countries who have been vaccinating for HPV since the early days of the vaccine, for example, in Australia and US, the level of the incidence of cancer, cervical cancer, is actually reducing. Now, my question is, since HPV is also associated with a number of other malignancies, head and neck cancers, penile cancers, and so on and so forth, is there any evidence that those malignancies are also reducing?
Not yet is the short answer. Esophageal cancer is reckoned to be the cancer which most commonly after cervical cancer is induced by papillomavirus infection. The problem there is that the time between the infection and the development of an obvious cancer is thought to be between 30 and 40 years. And of course, unfortunately, we've only been vaccinating in Australia against papillomavirus infection for 17 years now. But studies are now in progress looking to see if there is an esophageal precancer lesion that we can identify that might have gone away. It's not so easy in the esophagus as the cervix. The cancer where we probably will see an effect relatively soon will be in vulval cancer. But vulval cancer is quite rare, and the statistics gathered on it are not as good as for cervical cancer. So we're again studies have been set up to specifically look at whether we're getting rid of vulval cancer. But I think it's going to be about 20 to 30 years before we have the answers for the other cancers as clearly as we now have it for cervical cancer.
But Ian, did you actually see a future without HPV? In other words, we managed to eliminate or eradicate smallpox. And I know papillomaviruses exist in various other animals, but the human papillomavirus is specific to humans. If the vaccination coverage was, as we would like it, virtually across the board around the world, do you think, is it possible that we could see a future without HPV totally? Eradication, in other words?
I think we'll get rid of HPV 16 and 18 because the first and most important, the papillomaviruses are double-stranded DNA viruses. They do not mutate. So we have not got any evidence at all that there would be immune selection pressure on these viruses. So I think that the vaccines will be effective against 16 and 18. The problem, of course, is that there are lots of other papillomaviruses. The cervix, in the cervix, there are certainly upwards of 30 known papillomaviruses, most of which don't seem to cause cancer. So we won't get rid of the other ones, but with a bit of luck with the vaccine that we've now got, which is 7-valent, we should be able to get rid of the common cancer-causing ones. And yes, I would like to see in my lifetime, but I suspect it will be in my children's lifetime, that we can get rid of HPV 16 and 18, for example, altogether.
There's a question from the audience. To what extent can we predict the effectiveness of immunotherapies for various patients and ethnicities and genetic features? So basically, in terms of the geneticity and the variation, is there any comment on that effectiveness?
Yeah, it's a good question. And I think that the answer will be yes, because we recognize that our ability to make all sorts of immune response, and specifically T-cell responses, is very much genetically determined. So that you could argue, for example, that the patients who get HPV infection and do not get cervical cancer are the fortunate ones that have got the right genetic background to get rid of the virus, and that it's only the patients that have got the wrong genetic background that go on to get cervical cancer if they get HPV infection. So that that's not a proven statement, by the way. There is evidence supporting it, but what it really says is that we know that there are human MHC types, antigen-presenting types, that are more likely to get rid of HPV, and there are types that are less likely to get rid of it. And there's a lot of work being done to look and see whether there are other factors that determine who keeps it and who gets it. But I would predict that checkpoint inhibitors will be more or less effective in different people according to which particular set of checkpoints they naturally induce in their tumors. Because we recognize that the last time I counted, there were about 16 separate checkpoint molecules that can be induced on a tumor. And I'm sure that all of them will be differentially regulated by different genes in people that are more or less prone to getting particularly infections, because obviously the real purpose of checkpoint molecules is to switch off your immune response when you don't need it anymore. Obviously in a cancer, you don't want it to switch off, but in most people, it's efficient to switch off the checkpoint molecules when you've got rid of the infection that induced them.
Another question here is that, can you provide some insight into the potential long-term side effects of cancer immunotherapies and how researchers working in the field are trying to mitigate them?
Yes, it's obviously a challenge. You don't switching off the immune system. Switching off the immune system is something that you have to do at your peril. And obviously, we're manipulating a significant part of the immune system to try and encourage particular sorts of immune response. And in the process, we'll almost certainly be switching off others, so that we really are going to have to keep a close eye on what works and what doesn't. But the practical reality is that for many cancers, the only therapies that we have that are effective, if you can't cut them out with surgery, are now checkpoint inhibitor drugs. And I think we'll learn the hard way what the long-term consequences of administration of those drugs is going to be. We certainly have got a study running in the hospital where I work, where every patient that gets a checkpoint inhibitor is enrolled in a long-term study to see what's happening. And I'm sure that that will be happening worldwide as well. We would really like to be more selective about which checkpoint inhibitors we use, because we're at the moment we tend to use the ones which are most easily available commercially, but we know that there are many different ones that we haven't yet really mapped out as to regards to efficacy for particular cancers. And we will also have to look out for the potential side effects, because if you're basically allowing an immune response to run, you would expect to see autoimmune diseases appearing as well.
There's a nice question here from Lawrence Young from UK. What about the immune responses to E6? Would including this in the vaccine along with the E7 produce a more effective anti-tumor response?
Yes, I think it would be fair to say that we are using E6 and E7 for our studies, and I think most people are. We did the work on E7 because we couldn't show any direct effect of E6 on the immune system. E6 is a pretty powerful regulator of cell growth and division, but we couldn't find any direct evidence that E6 was doing something to the immune response, whereas for E7 it very clearly was. So I'm not saying it's not important to use E6 and E7. I would put E6 and E7 into any vaccine because we know both of those proteins are expressed in tumors that are induced by papillomavirus infection. So that makes perfect sense.
Similar lines, I mean, combining mRNA vaccine with immune checkpoint inhibitors is proving to be effective in melanoma and other cancers. What about this approach in treating cervical cancer?
Yes, I think that it will play a role in treating cervical cancer. I mean, the problem with cervical cancer is that, of course, we have quite effective treatments for most cervical cancer in the developed world, and it's really only in the developed world that we could talk seriously about inducing some of these more exotic treatments. But for the developing world, we really have to concentrate on preventing the virus infection. And I think that we could become very much more adventurous with cancer immunotherapy in the developed world, but at the moment the cost is rate limiting even in the developed world.
Another question here is, are there any specific biomarkers or genetic markers used to predict the response to immunotherapy in cervical cancer caused by HPV?
The short answer at the moment is, to my knowledge, no. But I'll be pretty sure there will be some. The only one that we have at the moment that got some robustness to it is MHC typing, which does predict whether you'll make a strong immune response to E6, E7 proteins. That's worked out over studies which have involved tens of thousands of patients. So mapping further potential predictors of outcome to particular sorts of immunotherapy is going to be a serious challenge for the future. I think the message from all of this is that we have to treat every patient that takes part in any study of immunotherapy, not only as a patient, but also as a part of a study, so we gather the data. At the moment, we phenotype the tumor, we phenotype the patient's immune response, and we keep collecting these data and sieving and looking to see what we can find out, in the hopes that we can get markers which will tell us what would be right. I think it's not feasible to test them one at a time, but it might be feasible if we just blanket test everything that we can in the hopes that we will get some answers out. And I think that's now affordable. I mean, given that we're the amount of money that we spend on healthcare, the extra cost of phenotyping individuals is actually not that high. The problem, of course, is that we've got to then look after all these data in some useful way, and that really will depend on having good facilities for storing patient data and permission from the patients to do so.
Another question here is bringing in AI. So the question says, given the potential of advanced artificial intelligence to unravel biological mysteries, how might the super intelligent AI system dissect the interplay between staphylococcus bacteria and human skin cells at the quantum level to elucidate the mechanisms driving skin cancer development?
Yeah, it's a good idea. I mean, we are sort of nervous about using artificial intelligence at the moment. We really haven't defined yet just like how well it can give us back anything that we didn't tell it to give us. But it does lead to the potential for smart further experiments to test ideas that an artificial intelligence program might come up with. The boundary between what we're doing on spatial transcriptomics at the moment and artificial intelligence processing of those data is pretty narrow. We really do already use effectively artificial intelligence to sift through these data sets. It's what we haven't done is teach the artificial intelligence all we know about all the other conditions where the immune system is disturbed, and where that might give us some clues, or give the artificial intelligence some clues where to look for things for cancer immunotherapy. It's a good idea, and I'm sure it will be being done. The rate at which we're using artificial intelligence just for diagnostic purposes is increasing steadily at the moment.
Mm-hmm. Another question, a brief question, but direct to the point. What is the reason for the age restriction for the HPV vaccination?
Well, there is absolutely no age restriction on medical grounds. We know the vaccines are safe. We know that they work. There's not much point in giving a vaccine to a 4-year-old to protect them against a risk which won't occur till they become sexually active, because the immune response will probably wane over time. And we really don't know how quickly the immune response will wane. We know that there is protection out to 10 years, but we don't know how long beyond that it's going to last. And then, of course, at the other end of the age spectrum, if you've already got the virus and got rid of it, then you're probably clear. And if you've got the virus and haven't got rid of it, then that will show up with testing for HPV, and the vaccine won't help you because it doesn't cure, it only prevents. So that I think that the age limits that are put on the vaccine are pragmatic to try and focus the use of the vaccine on the age groups that will get the best benefit from it. And certainly globally, that's what we should be trying to achieve. We want to get young women before they become sexually active, and we want to make sure that those vaccines are made available up to an age where most people will already have been sexually active.
Ian, on a similar sort of note, obviously there's a lot of, if you like, talk about, and to some extent this is being implemented in numerous countries with regards to vaccinating males as well, boys as well, kind of thing. What's your thoughts in terms of sort of mass vaccination of boys as well, similar to girls across the board? And I mean, there's differences in terms of opinion, people talking, because obviously, as I say, it takes two to tango. So vaccinating boys, yeah, go on.
Yeah, look, it's interesting. If you model the thing, then if you get 80% of the girls, that's pretty effective at controlling without doing the boys. But if you only get 50% of the girls, you need to do the boys as well. In Australia, we vaccinate girls and boys equally. Many countries do the same. That seems sensible to me. It increases the efficacy of the vaccine program, because you never can say that you've immunized every potentially at-risk girl or woman. And men get cancer from these viruses too. And while we don't absolutely know yet that esophageal cancer is prevented by vaccinating boys against HPV, it's a pretty likely event that at least some of them will be, because we know that they're driven by HPV 16 when we actually look at the molecular basis of the cancer.
It's gone 11 o'clock, Ian. Just one last question in that case. I apologize to all the audience where we've not been able to address all of the questions. There's too many questions to be in the limited times we have. And I know that Ian's got another session to go run to in a while. So just last question, what do we do with regards to vaccine hesitancy? In your slide, you mentioned that some of the countries which really need the vaccine is in the African continent. And those, from the best of my understanding, those are the countries where you get a fair bit of hesitancy in vaccination in the first place. How do we overcome that?
Well, I think that it always comes down to education. It's interesting that you need to have role models. I mean, within the African continent, the First Wives Club, the wives of all the presidents, they get together at the papillomavirus meetings and talk about what they should be doing about encouraging vaccination. I think that's a really smart idea. Leading by example is clearly the way to do it. People are naturally suspicious of things they don't understand, and therefore education about health generally and about what we know about vaccines specifically is a good idea in schools. We do it in Australia. I'm sure we could do it and encourage it to be done in other countries. One of the big problems is that for many countries, you get contact with a child at birth, first couple of months, and you don't see them again until they have their first child. You know, the continuity of care in childhood is not great. And that's one of the things that the World Health Organization recognizes, that we need to increase mother and child health by making sure that all kids get looked at appropriately during childhood to prevent preventable diseases like iron deficiency, for example, which is relatively common in some countries. And vaccination clearly is a part of that sort of program where we really need to make sure it's rolled out effectively. It's what the WHO is for, and they're funded to do it. And I think that they have the capacity to do it, but they do need the support of each of the countries where they want to go and work.
With that note, Professor Ian Fraser, thank you very much for a very enlightening, very enjoyable talk. Thank you to the audience. Thank you to the Sheikh Hamdan committee, organizers for this wonderful event. And I believe the seminar was recorded and actually live streamed on YouTube, on the Sheikh Hamdan Awards YouTube. So it will be available. Those individuals who either joined late or were not able to see the video and the seminar, they can go and see it on the YouTube channel. Ian, once again, thank you very much. It was great seeing you. Speak to you soon. Take care, everybody.
And thank you very much. And thank you also for the award which you gave me, which was very much appreciated.